Thank you. Thanks for the nice introduction. So I'm going to talk about today again, maybe we have heard already a lot on that, but mRNA drug formulation for genetic diseases. Why mRNA as therapeutics? Because they have high selectivity for their targets, and they allow a transient transfection, meaning that there is no permanent alteration of the genome, and they only need to reach the cytoplasm, and they are degraded via physiological pathway. Well, they have also implication, mRNA delivery implication, because they are negatively charged and they can be degraded by extracellular RNAs or endonuclease or exonuclease. So therefore, there is a need for delivery vehicles. I think that's what we have been hearing so far. So those vehicles can be viral vectors, but also non-viral vectors, and it can be synthetic, such as the lipid nanoparticles that I'm going to talk more today, or natural as the exosomes that we have heard also during this conference. And then when you really have this nice system where you have your cargo loaded into the vehicles, you need to process the delivery. And this is, again, a multi-step process. So, of course, we want to reach to target the appropriate organ and to reach the correct tissue or cells. You want to have good cellular uptake. You want them to escape the endosomal compartment and reach the cytoplasm, and to induce good mode of action in the cytosols. So with this, actually, rather than the development of or the delivery of vehicle properties, it's the insufficient understanding of endosomal escapes that is today a main limiter of the cytosolic viability and activity of the nucleic acid in the cells. So with this, it's estimated that it's around 1 to 2% here that actually the mRNA reach the cytoplasm and escape from the endosomes. So with this in mind, the research center has been funded. It's called Formulations. It's academic Swedish partner, but also private Swedish companies, and with the general aim to design new lipids nanoparticles, formulation, and that gain inspiration of the nature's use of extracellular vesicles and exosomes to transport nucleotides such as mRNA. So we are, as I said, several partners, and we have organized the work in different subgroups, but with two main challenges. One is to elucidate the critical molecular and cellular factors for a successful uptake and cargo release after endocytosis. And then with this, we aim to develop more innovative drugs for vehicle concepts that are efficient and safe for clinical use. So this is the outline of my presentation today. I will talk about the LNPs that we formulate and present some characterization study or data. Then I will show you some data that were made with confocal microscopy, flow cytometry to monitor cellular uptake and protein expression, and finally some intracellular trafficking data. So for the formulation, we use actually the precision nanosystem device that is here. They also have a stand here, so you can go and talk to those people. So basically, it's a microfluidic system. So with two inlets on the ship, then on one side you put your mRNA in usually aqueous buffer. On the other side, you have your lipid mix, and then you push everything into the ship, and you get your lipid nanoparticle here, pretty pure, that just need to be dialysis and to get pure compound, or actually you change the buffer here, and you have something that is at physiological pH. So the structure has been described recently in a PNAS paper, where actually we have inside in the core, we have the mRNA that is trapped with the cationic lipid MC3 and the cholesterol. And then we have first layer that is here in yellow, where which is enriched in a DSPC, and then an external layer where we have the PEG 2000. What is interesting here is that we know that by playing on the amount of DSPC, we can change the surface composition, and by playing on the amount of PEG or DMP-PEG lipid, we can change the size of diameter size of the LNPs. So that's what we have been doing, actually. So we formulated 3 different nanoparticles. This one that is called MC3M, let's say M for medium, with this lipid composition here. Here, as I said, by reducing the amount of DMP-PEG, we could enlarge the diameter size, keeping the surface composition constant. This is KSC. And then if you change, both reduce the amount of DMP-PEG and increase the amount of DSPC, then you change the size, but also the surface composition, which I call here variable surface composition. So those nanoparticles were encapsulated with or loaded with an mRNA that has a Cy5 tag and fluorescent tag, and this mRNA encodes for the protein EGFP. So here's some characterization data done by NTA, where we have the size distribution here and the particle per ml concentration. We also did some DLS measurement to make sure that we have something that is that correlate and validate. The Rebo green assay is used to determine the amount of mRNA that you encapsulate in those LNPs. And here, it's as you get usually with this system, it's around 97 to 99% of mRNA encapsulation. So with this, what we were interested in the role of the size and the surface composition of this LNP for cellular uptake and protein expression. So these are confocal microscopy data that were done on U7 cells, which are hepatic cell lines. So the signal was monitored here over 17 hours, and the red signal that you see is the Cy5 signal. The signal in green that you can see, of course, is the production of the GFP proteins. So it's playing again, I hope. No. Okay. Just to show you that. Here, no, okay. Can you play it again, or? Okay. Anyway, it's also to maybe you had time to see it, but the smallest nanoparticles are internalized more rapidly compared to the big nanoparticles, and it seems also that the one on the bottom right here, which is the larger one with the constant surface composition, seems to be more efficient in producing the GFP. Now it's coming, but somehow it's paused. Okay. So after this, we went for a flow cytometry measurement to quantify these signals, actually. So this is the Cy5 signal that is showing here for the 3 different LNPs. We do see after 3 hours that actually the small one is internalized faster by the cells than the larger one, and then if you change the surface composition, then you reduce the uptake. If you look at other time points, such as 6 hours and 24 hours, then actually it's the larger one that is the most efficient in being internalized. Then if you look at the EGFP signals, you also can see that it's the biggest one that is more efficient in producing the GFP protein. So now this is the workflow for intracellular trafficking study, and we so it's two different approach here. What we do first, we take some cells that we overexpress with the DNA plasmid that encode for organelles markers. So Rab5, it's very known for early endosomes, or then we use the Rab11 for the recycling endosomes, Rab7 for the late endosomes, and LAMP1 for the lysosome, for instance. So one way is to overexpress these organelles markers and then to expose the LNPs and go for kinetics, endosomal escape event, and colocalization. And we aim to reproduce such a diagram here in living cells. Another approach that we are doing in parallel is to establish the CRISPR knock-in cells, where we actually have more physiologically relevant Rab5 expression, and not only Rab5, but we started with this one. The aim is to also go for the other organelles. And before, when you have your cells and before going for your LNP exposure, then you need to do some validation of your models, and this go through endocytic capacity measurement, or also the number and size of the organelles, and this you compare it to the wild-type cells or the one that are overexpressed with the plasmid. So this is preliminary results that we have. Where actually we use the Rab5 GFP here, so don't make the confusion. It's in green this time the Rab5, so the early endosomes that are labeled here. And upon exposure to the LNPs loaded with the Cy5 mRNA, then we could catch, as we were expecting, the Cy5 inside the early endosome here after 2 hours exposure. And here on the right, it's another movie. Okay, great. So where you see that here the cells were overexpressed with the different organelles markers, and we have the LNPs again loaded with the Cy5 mRNA that encode for the GFP. So it looks a bit complicated, but over time actually you can track the colocalization, and as I said, you can see intracellular trafficking of the LNPs inside the cells. So you need to reproduce this and to have much more of a lot of data and to process them and to get something that is relevant and that you can quantify. One thing that is nice that we have observed here is that we can also monitor the mRNA release by steady-state emission measurement on a SPECS system. So we noticed that actually the Cy5 mRNA has different spectral signature, whether it is encapsulated into the LNPs or not. And this was done with the different amount of Triton X here to provoke the release, and then we could see actually the shift from the black curve where you have your mRNA Cy5 inside the LNPs to the green one. So this shift, we hope to use this signature to also catch endosomal escape events. So some summary here. We just discussed that the different LNP size or surface composition have different uptake kinetics, but also abilities to drive protein production. Intracellular trafficking study, so we get with this information on the mRNA distribution upon LNP exposure, and we are also establishing the CRISPR knock-in cells as new tools for, again, more physiologically relevant models. And also just to mention here that we are doing work on elucidating the uptake mechanisms, but also developing live cells imaging assay to detect endosomal rupture events. So with this, I would like to thank Eline Espionner-Winter from the PI of the group, and two of my colleagues that were involved, are involved in this project. Our colleague from AstraZeneca and all the fundings and the CLINAM organizers, and all of you for your attention. Thanks.
Thank you very much for this very interesting and very nice talk. So we have time for some questions.
First, very interesting talk, congratulations. And second, I haven't seen if this experiment were carried out pre-incubating nanoparticle in serum.
You mean pre-incubating the nanoparticle in serum to create a protein corona and then change the uptake? Yeah, that's something to consider. Now we are using 10% of serum in our system. We have done some work with using 1% of serum, and we do see some differences. So that's something to consider. I would say more for the uptake mechanisms and not really for the intracellular trafficking. Or maybe yes, but you never know. Yes, that's something to consider.
Just wonder on the background. When you show us that the RNA is actually embedded, not embedded, encapsulated, is it really the real physical picture, or is there some interaction between the lipid and the RNA in the way that it is packed? And if this is the case, maybe there are ways to optimize the system by changing the makeup of the lipid.
That's the confirmation. It has been observed, and this is you have more description on the paper, on the PNAS paper, and that's some confirmation that has been characterized. It's hard to say if it's the real structure or just interaction, but yeah.
Thank you very much. It was some great image, microscopy image. My question is that if you want in the future to be able to observe single RNA molecules, I guess. So how do you plan to do that? Can you just elucidate us? Thank you.
Yeah, that's a good question. Before going for single mRNA molecules, we also are interested in single lipid nanoparticle, because once they are internalized, then there are several into these endosomes and so on. So then the plan now, or what we have been thinking, is to change the way you expose your cells. Rather than doing low dose and continuous exposure, then we would go for a low short time exposure with a higher dose. So then already you remove a lot of LNPs in your system that can enter after the first one. Then you have continuous entrance. So that's one thing to make it a bit more simple. And then single mRNA. Well, that's another thing. What we were thinking is, or what we are doing actually, it's we develop tools using mRNA binding proteins, and we have another group where they focus on synthesis of mRNA, and then we aim to generate a thread between the species, and then to check for individual interaction.
Question at the back and then in the middle.
Hi, I was wondering if you check the stability of your nanoparticles in complex media, because, for example, I can imagine that in higher concentration of serum, your particle stability might change. Also, the size of your particle and the charge and the surface characteristic of your nanoparticles can change. So have you checked this?
No, not in serum media. What we have been discussing this earlier with another person, but so it turns that the LNPs are very stable. Like after 2-3 months, you still have LNPs with the same size and so on. What is a problem is the stability of the mRNA once it's encapsulated into the LNPs. So here we have some problem, and we have seen that we need actually to use those LNPs with the mRNA when they are freshly formulated. If you wait for 2 weeks, then you do see differences in the protein expression. So stability is something that is done on parallel, but for this method development assay and tools, we just formulate fresh LNPs and then we go for our experiment. We don't keep the LNPs and use them too long.
Hi, thank you for the talk. So I'm just wondering, have you tried doing the same sort of work on natural systems, i.e. the exosomes, and do you see, if you have, then do you see similar or different sort of intracellular trafficking upon intake?
Yeah, that's a good question. That's the goal, or one of the goals of this project. So just to tell you that this Formulation Center has just started end of next year, and we have a group at Karolinska Institute in Stockholm. They are making exosomes and trying to establish a pairing between cells. Our job at Göteborg at Chalmers is also to use our tools with their exosomes and to, as you said, track intracellular trafficking. So this is not done yet. This is something that is planned.
You are very popular. So I have the last question.
Yeah, sure.
So it's just a technical question. I mean, you showed, if I remember correctly, that you had 3 types of particles. One, a small one around 70, 80 nanometer, and then the other 2 were similar, about 120-ish.
Absolutely.
And then you showed that the encapsulation efficacy was technically 90% across. And you also said that the bigger particles were taken up better into the cell. Now, the question I have is, when you compare the smaller one versus the big one, I mean, in terms of a spherical particle, it's a considerable difference in the volume, right? So what is the absolute quantity of the messenger RNA per vesicle? I mean, how many messenger RNA do you have per small particle versus the bigger particle?
Yeah,
so that's the first question.
I understand your point, yeah. I don't know exactly how many molecules we have, but this is always the thing when you work with the delivery system. Right. They have different properties, so the encapsulation can be different. And then if they have different size, then it's always a matter to how do I express my concentration in terms of number of particle per cells or in terms of massive concentration. So there is always this gymnastic to think about. So in our case, what we have similar, as you said, encapsulation rate. We have different sizes, but we also have a similar particle per ml concentration.
Right. Because that brings me to the second part, because it's related in terms of what is it that you are measuring. Because if, let's say for the sake of argument, you may have, let's say, 1 or 2 messenger RNA more in a bigger particle than the little one. Then you are measuring the fluorescence. So you are dealing with a quenching problem, which you may have less quenching in a bigger particle, but you have a lot of quenching on the smaller particle, which gives you the impression that you have less uptake.
Yeah,
so
that's
how do you correct for that?
That's a good comment. We don't correct for that so far. The PNAS paper I mentioned, this study was done with the mRNA encoding for the EPO. It's a protein that is secreted, and we used exactly the same same system, even more actually. But those 3 systems are also there, and we measure by ELISA assay the protein that is excreted, and we do see the same. We can rank the particle efficiency in the same way.
Okay, thank you very much.
You're welcome. Thanks.